Donc je suis très ravi de revoir mon ami Eugene Koonin, avec qui on a beaucoup travaillé ensemble. Philippe Colson, j'ai envoyé Philippe Colson en postdoc chez Koonin. Et depuis, d'ailleurs, on en a collectivement un usage important. Et Eugène était un homme charmant, intelligent, vif, qui est un bonheur d'écouter en général. Donc je suis très heureux pour moi et pour vous que vous ayez la chance d'écouter Eugene Koonin.
Thank you very much for the kind introduction, Didier. And let me start sharing my screen without much further ado. Bonjour, good afternoon. Thank you very much again for the invitation. Unfortunately, I have never been physically in the institute. I really look forward to visit. But for the time being, virtually, I am pleased to talk to you about the great world of viruses and its evolution. I will not discuss SARS-CoV-2 or COVID-19 in this particular presentation. I could have done a different one where I would discuss the evolution of this virus, but I always think that in the era of the pandemic, hopefully in its waning days, it is important and surely interesting to get a broader perspective, greater perspective on the world of viruses. And I put here this saying from one of the letters of Albert Einstein as an epigraph. The fact that the world is comprehensible is a miracle. Indeed, there may seem to be good reasons why we sitting on this little planet should be able to understand something important about the world, but we do. And the same applies to the world of viruses. So let me just do something with the pointer and proceed. Okay, so by way of self-introduction, I run the Evolutionary Genomics Research Group, or lab, at the National Center for Biotechnology Information in Bethesda on the NIH campus. And as you see, it's not a small lab. Because of the pandemic, it's an outdated photo, but the lab is about the same size anyhow. And we do all kinds of theoretical and more empirical or even applied studies in evolutionary and comparative genomics. But in particular, one of my main interests, main fascinations is the evolution of the world of viruses in all its aspects. So, to begin with, I always show this slide, and this is one of the actually important take-home messages for those who may not be familiar with it. Viruses are the dominant entities in the biosphere, in all senses, physically, numerically, genetically. As in particular shown by viral metagenomics, by metaviromics, as we sometimes call it. The numbers are really staggering. 1 cubic centimeter of seawater, for instance, may contain, depending on the particular habitat, from a million to a billion virus particles at any given time. In many environments, the virus to cell ratio is between 10 and 100. So there are 1 to 2 orders of magnitude more virus particles in many environments, not all of course, but many, then there are cells. And the Earth-wide counts are truly hyper-astronomic. The rough, but apparently rather robust current estimate is that there are about 10 to the 31st virus particles at any given time on our planet. And it should be noted that the genetic diversity of viruses, which tend to evolve at a very high rate, is also commensurate with that numerical and physical abundance. So viruses are the main pool of novel genes on Earth. To do a little comparison, largely for fun, it doesn't have a particularly deep meanings. There are about 10 to the 24th stars in the observable region of our universe, and about 10 to the 31st virions at any given moment on Earth. It's when I say hyperastronomic numbers, it's not so much a metaphor. It's indeed what it is, about 10 billion times more. But that's not all. And what I'm going to say next is sort of textbook knowledge, but I want to emphasize it. Almost exactly, just a few months over half a century ago, David Baltimore, one of the discoverers of reverse transcriptase, presented this classification of viruses that has become highly popular. Dividing them into 7 classes based on the form of the encapsulated virus genome and accordingly on the cycle of virus replication and expression. All cellular organisms, you and I, bacteria, archaea, plants, etc., employ one and the same route of information replication and expression. Our genomes are represented by double-stranded DNA, which replicates but is also transcribed into a great variety of RNAs. One class of them, the mRNAs, are translated into proteins, and that's completely uniform across the entire diversity of cellular life forms. Viruses are very different in that respect. Basically, to put it plainly, viruses employ all possible forms of nucleic acids as genomes, which means the nucleic acids contained in the virion, and accordingly all ways of information replication and transmission. The simplest ones are, let us say, the single-stranded RNA viruses with a positive polarity of the genome that incorporate directly an mRNA or RNA of same polarity into the virions. Then there are double-stranded DNA viruses that incorporate double-stranded RNA into virions, and accordingly it has to be transcribed to produce mRNA. Negative sense, single-stranded RNA virus is the same. It needs to be transcribed to produce mRNA. Reverse transcribing viruses that contain either RNA or double-stranded DNA in the virion, but are united by the fact that they include a reverse transcription step in their reproduction. Single-stranded DNA viruses with small circular genomes, typically that replicate by rolling circle mechanism. And finally, the class that corresponds to the information transmission scheme that is characteristic of cellular life forms, double-stranded DNA viruses. So in a sense, from the informational point of view, we all belong in this single class of viruses. So this diversity of genetic cycles hints at the possibility that viruses are extremely ancient and might have been, so to speak, the original genetic test systems at the earliest stages of life evolution. This idea is compatible with the ubiquity of genetic parasites and the inevitability of their existence. So genetic parasites, and in particular viruses, are associated with all cellular life forms, maybe with a few exceptions of intracellular, parasitic, and symbiotic bacteria. And moreover, there are I think, very good theoretical arguments, into which I do not have the opportunity to go in detail now, that indicate the inevitability of the emergence of genetic parasites in all evolving life. So that is really well compatible with the notion that viruses have accompanied all evolution of life on our planet, might have even preceded the emergence of the first modern type cells, as we shall discuss in greater detail in a few moments. At the same time, it has to be actually adding to the notion of diversity. A crucial point should be mentioned right now. There is decidedly no common ancestry of all viruses, very much unlike cellular life forms, which are all monophyletic. Accordingly, the evolution can be studied productively through phylogenetic analysis of universally conserved genes, such as ribosomal RNAs. Nothing like that in viruses, not a single universal gene that could serve as that type of phylogenetic marker. So, I will now discuss the the old as well as new emerging ideas on virus origin. The subject of virus origins has been discussed in the literature for over a century, but until the last 2 decades, let us say, maybe even primarily 1 decade. These were speculative discussions. There was really no data to support one or the other type of scenarios. But now this situation has changed, thanks to genomics, thanks to structural analysis of numerous viral and cellular proteins. I think we have insights into the likely reality of virus origin, but let's review the scenarios first. To begin with the very beginning, so to speak, here's the virus early hypothesis to which I already alluded. Namely, it postulates that viruses come from the primordial pool of replicons that preceded the emergence of first cells. Opposite to this is what may be called regression hypothesis, namely that viruses as ultimate parasites descend from cellular parasites. I already mentioned intracellular parasitic bacteria that reproduce inside eukaryotic and in some cases even inside other prokaryotic cells. And so the gist of this hypothesis is that viruses are the ultimate form of degradation of such intracellular parasites. And finally, perhaps the most popular historically, escape gene hypothesis, according to which viruses originate from, so to speak, normal cellular genes that somehow have gone berserk and acquired replication autonomy. Now, let us say how we can harness empirical evidence to assess these different scenarios. As I mentioned, there isn't a single universal gene among viruses, which greatly complicates the study of virus evolution. Things, however, are not entirely bleak, because there are no universal genes, but there are about 15 or perhaps 20, depends on the exact definition, genes that are widespread among highly diverse viruses. I call them virus hallmark genes. Not unexpectedly, probably, these hallmark genes encode key proteins involved in virus replication, formation of virion structure, virion morphogenesis, and they're present in highly diverse viruses, some of them spanning 2 to 4 of these Baltimore classes that I described a minute ago. They're just shown here as the structural schematics, because structure are available for all of them, and these are capsid capsid proteins, single jelly roll capsid protein, double jelly roll capsid protein, as well as proteins play key role in virus replication, such as RNA-dependent RNA polymerase, reverse transcriptase, the so-called superfamily 3 helicase, and nuclease involved in the initiation of rolling circle replication, and a few other somewhat less widespread but still common among viruses hallmark genes. So by studying the evolution of these genes and their combinations, we can get insight into even into the very origin of viruses. And let's begin now with the genome replication modules, the proteins that are required for virus replication, such as polymerases, primases, and rolling circle replication endonucleases. These are very different proteins. If you do typical sequence comparisons, such as BLAST, or even more sensitive methods such as HH-PRED and HH-search, you do not really see similarities between some of these classes of virus replication proteins. For instance, there's between RNA-dependent RNA polymerases and DNA polymerases. You'll simply not be able to detect it. But when we perform structural comparisons, the picture changes. Actually, rather astonishingly, at least to me, underneath all this diversity and complexity lies unifying simplicity. Namely, at the core of all these replication proteins is the so-called RNA recognition motif fold, as you can see here on this schematic, on this slightly more realistic representation. It's a very simple protein fold that is found in a great variety of RNA and DNA binding proteins, as well as in many enzymes. The wide simplicity and widespread and functional diversity of this domain leaves no doubt that it comes from the very earliest stages in the evolution of life. Indeed, any reconstruction of the last universal common ancestral of cellular life places quite a variety of proteins with RRM fold into that LUCA, last universal cellular ancestor, implying that a lot of evolution of proteins containing this domain occurred at early stages of evolution. So a lot of diversification happened already by that time, which to me is a strong indication that this domain actually was one of the first protein domains to appear when life was transitioning from the primordial RNA world to the modern type RNA protein, nucleic acid protein system. And originally it might have been even a facilitator and stabilizer of ribozyme polymerases. But when eventually, in the course of evolution, catalytic residues have been grafted onto this domain, leading to the emergence of all these viral hallmark genes involved in replication. So I think we can be confident about very deep evolutionary roots of virus genome replication modules, probably going back to the pool of replicators preceding the origin of modern type cells. When we look at viral capsid proteins and compare their sequences and more efficiently structures to those of other proteins, we detect a very different type of picture. Namely, for nearly all major Virion components, and this comes from 4 years ago, and this has been now expanded, for nearly all major Virion components, we detect likely ancestors among cellular proteins, primarily, and that in retrospect should not really surprise anyone, primarily carbohydrate or nucleic acid binding protein, suggesting that unlike the replication hallmark genes, the genes encoding the principal virion components have been recruited by evolving viruses from cellular ancestors at different stages of the evolution of life. Taken together, these analyses suggest what I call the unifying scenario that I call chimeric. Chimeric, or maybe hybrid, I don't know. Chimeric or hybrid in the sense the different components of viruses have distinctly different origins, with the replication machinery evolving according to viruses' early scenario from the pool of precellular replicons, but virion components emerging later from recruited genes of cellular hosts. Some emerged at a very, very early stages of evolution, apparently perhaps before the last universal cellular ancestor, but still when cells were already quite functional, such as the jelly roll capsid proteins of icosahedral viruses. Others appeared at later stages of evolution. I believe that this is now the most parsimonious scenario of the evolution of viruses that is likely here to stay. I will now finish with the very interesting, in my opinion, subject of the origin of viruses and switch to our day, to matters more practical and no less interesting. And what I want to emphasize is that metagenomics, and more specifically metagenomic sequencing of viruses, metaviromics as we call it, resulted in the advances of metaviromics, resulted in what amounts to a tectonic shift in understanding virus evolution. Indeed, metaviromics has now become the principal source of the discovery of new viruses. How dominant the metaviromic discoveries are, we shall see in a few minutes. But for almost all groups of viruses, this is now the principal source of virus discovery. And accordingly, the International Committee for Taxonomy of Viruses, as a result of lots and lots of debate, has recognized that in a big step, for a taxonomic body, it's a big step, has officially recognized that new viruses and the new virus taxa can be identified by genomic, solely by analysis of metagenomic sequences. A big step, but an essential, necessary step. And this slide illustrates why this is truly an essential step. So here this plot compares the prevalence and abundance of different viruses in different stations sampled in the world ocean. So the prevalence is how often in different parts of the ocean you detect the given virus genome or its close variants, and abundance means how many reads on average or maximum correspond to this virus genome. And the colored circles here represent viruses with known hosts, isolated by traditional means, whereas the gray circles represent those discovered solely by metagenomic analysis. And you can see with much clarity that both the most common ones, the most prevalent ones, and the most abundant ones at this logarithmic scale, note this, the most abundant ones are completely unknown, unseen by the traditional approaches, only by metagenomic approaches. And this is, again, somewhat outdated slide. Nowadays, if I bother to update it, you almost would not see those colors, leaving us absolutely no choice other than recognizing that metagenomes, metavirons, are the principal source of novel virus discovery and the principal driving force for understanding virus diversity. I'm giving this talk on the margins of another meeting on virus taxonomy occurring in Oxford, where this is emphasized as strong as ever. Okay. So with this introduction, I'll discuss now quickly, but with a few details, the great expansion of the RNA or the RNA virosphere brought about by metaviromics and the emerging notions of the evolution and diversity of RNA viruses. I will not really touch the viruses with reverse that use reverse transcriptase, just classic RNA viruses. So when it comes to RNA viruses, it's crucial to recognize that all these viruses actually do have a universal gene, a universal protein, which is RNA-dependent RNA polymerase that is found in all of these viruses that replicate autonomously. There are some satellites that don't have it, but all viruses that replicate autonomously encode RNA-dependent RNA polymerase, which is understandable because in cellular hosts there is effectively no RNA replication, so they need to carry their own enzyme to catalyze this process. Accordingly, the only way to study evolution of RNA viruses comprehensively is to explore the phylogenetic trees of RNA-dependent RNA polymerase. This task is quite complicated for two reasons. One of which is that there are very many of those sequences. This is already a large number for any bioinformatic methods, but we shall come to much, much bigger numbers. And secondly, despite their concerned role in the replication of RNA genomes, virus proteins evolve fast. And so sequences are highly diverse. So we had to develop some fairly complicated iterative bioinformatics pipeline, which with a variety of amendments, serves us to build robust alignments of RNA-dependent RNA polymerases and then trees, maximum likelihood phylogenetic trees, based on these alignments. I will not describe it in detail, but it's useful to realize that this is a rather non-trivial bioinformatics task. So this is a bird's eye view of the tree that we got about 3.5 years ago. In this case, about 5,000 distinct RNA-dependent RNA polymerases. When I say distinct, I mean that we collapse minor variants. We cluster the sequences such that all the entire groups of closely related sequences are represented by one. So this is the most meaningful representation. And this is the bird's eye view of the tree, in which, at the time, about half of the sequences were from known viruses and half from metaviromic data. And this is a more meaningful view of this tree, which encompasses the entire known diversity of RNA viruses, and which fortunately contains 5 major branches, very well defined, stable to the sampling procedures, tree building methods, which now getting a bit ahead of myself, are recognized as phyla by the International Committee for Taxonomy of Viruses. So a brief tour across these branches. The first one here that is closest to the root, and the position of the root I infer by the separation of RNA-dependent RNA polymerases from the homologous reverse transcriptases of non-LTR retrotransposons and group 2 introns, highly concerned reverse transcriptases homologous to RDRP. So if you root the tree this way, this deepest branch includes the positive sensed RNA viruses of bacteria, Leviviridae, at the time the only known family, now we have many more, and their direct descendants in eukaryotes, some of which fittingly replicate in the mitochondria. Two of these largest branches include primarily positive sense RNA viruses, enormously diverse ones, with some intrusion of double-stranded RNA viruses. This branch 4 here includes solely double-stranded RNA viruses, again highly diverse, infecting both eukaryotes and bacteria, this group of ssDNA. And finally, this very strongly supported group includes negative sense RNA viruses that are so far found only in eukaryotes and primarily in animals and plants. I could not resist the temptation to map a few of the most notorious human pathogenic viruses on this tree to show, including, of course, coronaviruses, polio, hepatitis C, Ebola, influenza, and I could have more evidently, to illustrate for you primarily that these human pathogens are scattered among all branches of the tree, with the sole exception of this one, that include primarily eukaryotic viruses. I didn't put anything here in branch 4, but I could have put rotaviruses, which are important human pathogens. So, in a sense, studying virus evolution is a practical matter that allows you to better understand the evolution of human pathogens. So, an important utility of phylogenetic trees, any robust phylogenetic trees, is that we can use them as scaffolds to reconstruct the evolution of various biological features and the evolution of genomes. So we did that for positive sense RNA virus evolution, and what we see here is quite an interesting and clear-cut picture. And that is a picture of gradual complexification of virus genomes through recruitment of various proteins and domains, in particular independent recruitment of 3 varieties of helicases that facilitate replication of longer RNAs. That trend towards complexification of virus genomes comes to head in none other than coronaviruses that contain the largest, most complex RNA genomes of all, with a variety of domains. Now, 2 RDRPs, the only group of RNA viruses contain 2 RDRPs, 2 proteases, helicase, a variety, a complex of nucleases, which are all involved in genome replication, as well as the notorious spike protein and E and M and N that are involved in the formation of the virion, as well as several ORs that are omitted here that contribute to virus-host interaction. So, In this previous analysis, we obtained the grand scenario of the evolution of RNA viruses, but by no account does it exhaust their diversity. And in this work, published less than 2 years ago, what we obtained is striking, because we observed the doubling of the RNA virus diversity from the analysis of just a single, even if large, metagenomic sample from a rich habitat, the estuary of the Yangtze River in China. Such great expansion of the RNA virosphere from the analysis of a single metagenome strongly implies that the true diversity is huge and remains to be investigated. The other side of this coin, however, is that while the diversity is huge and there are a variety of groups of viruses with very interesting features, which unfortunately I cannot describe in detail, the great majority of this new diversity falls into already known major group, and in particular in one of these 5 branches. No new major branch has been discovered in this study, indicating that perhaps we already have the gross structure of the RNA virosphere, despite its enormous diversity. And now, a very, very recent, still unpublished work in which we analyzed a very large number of diverse metatranscriptomes, a huge collection. And what we got here is about 5-fold increase in the diversity of RNA viruses at all, remarkably, at all levels of taxonomy, from individual clusters, from sequences, distinct sequences, to classes, suggesting that indeed a lot remains to be discovered, because we discover not only new species, new genera, new families, but also new classes. On the other hand, what we also find that we only identified 2 new putative phyla, 2 new putative major branches, suggesting again that the overall structure may already be known. Okay, I'll now leave the RNA virosphere and give you a very quick tour of the rest of the virus world. To begin with the reverse transcriptases, for which we also build a tree like this, in gross features quite similar to the phylogenetic tree of RDRPs, that depicts evolution of RNA viruses. And the interesting feature here is that this tree combines viruses from 2 Baltimore classes. Those contain green ones here that contain RNA, single-stranded RNA in the virions, and these brown ones that contain double-stranded DNA. The key point is that the tree is well resolved, naturally allows classification of these viruses. Of course, there are also notorious human pathogens here, such as HIV and hepatitis B. Single-stranded DNA viruses present a really interesting picture. A picture that is a challenge, admittedly, to any classification taxonomic efforts. Because what we discovered is that these viruses evolved on several independent occasions, in a sense convergently through the same route of evolution, from small plasmids replicating by the rolling circle mechanism, combining with gene encoding a capsid protein that comes from RNA viruses. That route of evolution occurred perhaps 6 or 7 independent occasions and led to different groups of these single-sided RNA viruses that, however, all contain this RCRE denoted here, rolling circle replication, initiation, and nucleases. And moreover, some of these viruses in the course of the evolution gave rise to small double-stranded DNA viruses, papillomaviruses and polyomaviruses that also include important human pathogens. Finally, the largest part of the virosphere, the most diverse part of the virosphere, viruses with double-stranded DNA genomes. Here, we cannot build a single phylogenetic tree because there are no universally conserved genes. So we turn to network analysis, to analysis of gene sharing bipartite networks with these larger colored nodes representing genomes and these tiny black nodes representing genes, and you can connect genomes by an edge when they share homologous gene. And notably, this network has a highly modular structure, and these distinct modules, at least 4 of these, include 2 vast groups of viruses with icosahedral particles, one with so-called HK97 capsid protein and the corresponding package in KTPase, and these are tailed bacteriophages and herpes viruses infecting animals, and the other contains so-called double jelly roll capsid protein, completely unrelated to this one, which includes non-tailed bacteriophages, adenoviruses, and all the large and giant viruses that tend to replicate in the cytoplasm of eukaryotic cells. These are all both huge modules, including an enormous variety of viruses. And these smaller modules, finally, include very interesting viruses infecting hyperthermophilic archaea, as well as polyoma and papilloma viruses that I have already mentioned. So, time to start summarizing things. The global architecture of the virus world that has been rapidly translated and officially recognized as a megataxonomy of viruses, with the higher level, the highest level known as realms, and here 4 major realms originally recognized, and underneath these are kingdoms, phyloclasses, etc. So, Virion, typical RNA viruses and reverse transcribing viruses, unified by homologous replication enzymes, monodna Virion, single-stranded DNA viruses, and 2 major realms of double-stranded DNA viruses. A little later, 2 smaller, much more narrow realms have been recognized. The realm ribosi Virion, which consists of so far a very small number of viruses, including human hepatitis delta and its relatives infecting other animals. These are highly unusual among viruses, where small circular RNAs, resembling in many respects viroids, but encoding a virus nucleic acid protein. And the realm adna Virion, which includes rod-shaped and filamentous archaeal viruses that unexpectedly contain the A form of DNA in the virions. And I think the discovery and recognition of these, so to speak, minor realms of viruses predicts the future of the study of virus diversity. We are unlikely to discover very large new realms, but a variety of smaller ones will be discovered, and some are already, in a sense, known, but await recognition. So I will go through very quickly through the next few slides, because it's impossible to explain the details. And they're here to make the point that we produce based on phylogenetic analysis of the highly conserved genes, we produce detailed taxonomic schemes for each of the realms. Here they are. So at the deepest level, the evolutionary relationships in the virus world can be represented by this type of network, where I superimposed these monophyletic realms of, let's call them monophyletic realms of viruses, and the Baltimore classes that differ by the type of nucleic acid incorporated into virions and information transmission mechanisms. They're all brought together at the deepest level by highly conserved domains, in particular the RRM domain as well as jelly road domains. But the relationship is not trivial in the sense that riboviria, for instance, included the whole of 5 Baltimore classes, whereas Baltimore class 1, double-stranded DNA viruses, is split between at least 3 distinct realms. But it's important to realize that underneath the diversity of the virus world is a deep unity. So to progressively come to even bigger generalizations, we can define the virosphere as a part of the replicator space. Replicators, I think it's a very important conceptual definition. These are biological entities that replicate their genomes but lack complete metabolic pathways, in particular energy transformation. And viruses are the type of replicators that encode a capsid protein. So all these realms of viruses belong here in what we denote ortho virosphere, typical viruses, which is embedded within what we call peri-virosphere, which contains derivative of viruses, for instance, capsid-less derivative, and all these virospheres are immersed in the broader replicator space that includes plasmids and transposable elements, and even conceptually cellular genomes. Okay. Before I finish my lecture today, I want to dispel an impression that might be emerging from the previous slides that I have shown, namely that we already understand most of what we need to understand about the world of viruses. Not so. Major new groups of viruses remain to be discovered. And the striking example on which I will spend just a couple of minutes are the most abundant human-associated viruses. So the most abundant human-associated viruses are not any pathogens, or not even any commensal viruses that infect our cells, but bacteriophages. And in particular, these bacteriophages, known as crAS-phages, that have been discovered by cross-assembly of DNA from human gut metagenomes, hence CRAS. So think about it. This is totally striking. These viruses in many of us correspond to 90% of the virus sequences, virus reads in the gut metagenome. However, they have been completely unknown until the advances of metagenomics. Since when they have attracted attention, they could be assembled only by careful analysis of metagenomes. Since when they have attracted attention, some have been cultivated, but before the advent of metagenomics and metaviromics, we simply had no idea of the most abundant viruses in the human body. Another interesting aspect about these viruses was that at this original analysis 8 years ago, over 80% of the predicted proteins encoded in the approximately 100 kb genomes did not look like anything, so the whole genome pretty much looked like terra incognita. Fortunately, a more in-depth analysis of protein sequences using more sensitive approaches changed this situation and predicted the structures and functions of many of these proteins, and no less importantly, greatly expanded the diversity of these bacteriophages, showing that this is not just one phage that is abundant in our genomes, but a large family of viruses, where these highly similar ones represent just a small branch. Many of them are associated with animals, but there are some environmental ones, and they're united by some signature of genes, but highly diverse already on their own. Very recently, we expanded this even much further, discovering thousands of highly diverse phages that belong to this group that in all likelihood will now correspond to at least an order of viruses, or maybe a class, and that possess a variety of highly unusual biological features. I think I come to my conclusions and stop here. In the conclusions, I don't need to spend much time on this, because I have already said all these things. Viruses are the dominant entities in the biosphere. Metagenomics and metaviromics are now by far prevailing approach in virus discovery. Simply, virus discovery is primarily done by metaviromics and metagenomics now. The diversity of viruses is still far from saturation, but most very high level taxa at the level of phyla are probably already defined. We may be settling on the overall stable scenario of virus evolution and megataxonomy. On somewhat smaller scale, however, a variety of abundant and extremely important viruses remain to be discovered, as exemplified with the crAS-like phages. And as for microevolution, I decided to exclude this from today's talk. That would have been too much. As I said, I could have given a whole lecture about SARS-CoV-2 evolution, etc. So contributors, collaborators with 3 colleagues have been most instrumental in this analysis: Valerian Dola from Oregon State University, Mart Kropovich from Institute Pasteur, and Yuri Wolf, a senior scientist in my own lab at the NCBI, as well as a variety of other colleagues from my lab and from other institutions all over the world, in particular this very large RNA metagenomic projects, which was led by very talented graduate student Uri Neri from Tel Aviv University. Thank you very much for your attention, and I will be more than happy to answer your questions.